Hey, I'm Mr. Williams, and this video is about RNA and protein synthesis. Before we get started, you probably should take good notes. Personally, I like outline style, but I incorporate it into a Cornell format. But you ever use whatever gets your dog to walk, so you do you. Make sure you're asking good questions in the Ed Puzzle, or you ask me in class if you have questions. Let's get started. When we first left the last video, the last thing we talked about was the three types of RNA: messenger RNA, transfer RNA, and rRNA. And in reference to this, we talked about what did Watson and Crick need to know. They knew that DNA needed to do it. It needed to be able to store information, to be able to replicate itself, be able to make mistakes, and it had to be able to be read. We last talked about how to read it. It has an alphabet. It's only 4 letters long: A, T, G, and C. It only has every word in its vocabulary. It only has 3 letters, a codon. As a result, there's only 64 words in the language of DNA. And then the last thing we talked about was, like I said, RNA, messenger RNA, transfer RNA, rRNA. We haven't talked about protein synthesis. How do we get the message, that the code that DNA stores to make protein? How do we get it out to the ribosome, and how is it changed into a protein? That's what we're going to talk about today. This one is going to take a little bit of time. You need to pay attention. All right. What is the role of RNA? Well, DNA codes for protein. That's all it does. And there's a sequence of events that takes place during protein synthesis. So what does RNA do for that? Well, it plays the part in both of these. These are the sequence of events. There's transcription and translation. And RNA plays a role in both of those. The problem is DNA is in the nucleus. The ribosomes are out in the cytoplasm, either attached to the endoplasmic reticulum, or if we're making proteins for outside the cell, or if they're for proteins inside the cell, then they're just free floating in the cytoplasm. But the DNA can't get to the ribosome because DNA is in the nucleus, ribosomes are out in the cytoplasm. So we have to have a way of getting that code out. That's the function of mRNA. Remember, mRNA stands for messenger RNA. All right? There's an enzyme. We'll call it maybe DNA helicase. You do not need to know the enzyme. Don't worry about it. But it unzips the part of the DNA that we're trying to get the code out of. So DNA, again, a 6-foot strand of DNA, 200 trillion base pairs. Only a small part of it codes for the protein that we're trying to make. So there's an enzyme that unzips that part of the DNA, and so we can read it. All right, messenger RNA, just like DNA replication, is made from a template of that DNA. All right, messenger RNA uses the C's and the T's, the A's and the G's in DNA to copy itself to make RNA. Except there's no thymine. There's uracil. So instead of A, T, G, and C, it's A, U, G, and C. You need to remember that. Again, a great test question. Like I said in the last video, if you saw this, that's a U. Is that RNA or DNA? You know it has to be RNA because they're using it. If you saw a T, it would be DNA. Make sure you remember that. So transcription takes place in the nucleus, all right, and looks something like this. Now, I'm not sure, honestly, I've got to be honest with you, I'm not sure this is important, it is accurate, and honestly, I don't care if you know that or not. We're talking about enzymes, you don't need to know that, but I'm not sure that's correct. There's an enzyme, we'll call it, in this case, they've labeled it RNA polymerase. RNA polymerase comes in and unzips the DNA, just like a zipper. The RNA, there's a enzyme. Actually, that's what RNA polymerase does. It comes in and it matches up the bases. So, and you can see the code down here. This is guanine, and on the other side is cytosine. This is cytosine, and on the other side is guanine. This is thymine over here, so we put an adenine, we're pairing it up, A's with T's, G's with C's. Straight line, crooked line, crooked line. But all of a sudden here, This is adenine. It should pair up with thymine, but it doesn't. It pairs up with uracil. So you're unzipping the DNA and you're just basically replicating it, but you're not replicating it, you're copying it onto RNA, with the exception of, instead of thymine, we're using uracil. That is transcription. It takes place in the nucleus. You probably ought to write that down. What is transcription? Well, it takes place in the nucleus, and it's copying the code from DNA into a messenger RNA. It's the same as replication, except that in RNA, there's no thymine, there's only uracil. Write it down. You got it? Okay. This new strand of messenger RNA can now leave the nucleus and travel to the ribosome. DNA can't leave the nucleus, but messenger RNA can. It carries, I don't know, what should we call it? We could call it a message. We could call it the message from DNA to the ribosome. Messenger RNA carries the code, the message from the DNA to the ribosome with this correct sequence of bases. Then once at the ribosome, we can read the codons. Remember codons? The vocabulary of DNA. We can read 3 base pairs at a time. It's read by the ribosome, and each amino acid is paired up with the correct codon. All right? But there's a problem. We need to, if you have 64 words in your vocabulary, all of our words have meaning. As I'm talking to you right now, as I'm saying things, you understand what I'm saying because you know the vocabulary. Well, we need to know what the vocabulary is in DNA, and we can do that by looking at this table. Oh my God, there's a lot there. You're right, there's a lot there. You do not need to memorize this, I promise. Matter of fact, you have access to this. It's in the study guide. It's, I think, I'm not sure, it's also in the Brainscape. But if the way you read this is you look at the first letter, remember a codon, it's 3 letters. So if I'm making a codon up here. Let's say it's G, C, A, C, A, G, okay? C, A, G. Well, the first letter is a C, all right? So it's going to be in this portion of the table. Oops, that portion of the table right there. The second letter is an A, so it's going to be in this one right here. And the third one is a G, so CAG codes for glutamine. Do you need to know that? No. I do not expect you to memorize this. I expect you to know how to read it. All right? Some students don't like this particular table. They like this one better. So again, it's C. Okay, I got to go back. What was it? It was CAG. Forgot. CAG. So we start with C. We go out to A. We go to G. And it's glutamine. You just work your way out. The first letters of the codon. The first letter of the codon is the innermost circle. The second letter is the second circle. The third letter is the outermost circle. Right? If you like this one, then I would recommend you go to because I don't give you one of these, I would recommend you go to the internet and print yourself out one that you can refer to. This is I think it's actually a slightly better table than the first one because it's more clear. Here's the thing to think about. There are 64 codons, but there's only 20 amino acids. So some of those codons code for the same amino acid. And you can see that right up here. Like UCU, UCC, UCA, and UCG all code for serine. Do I expect you to know them? No. If I give you something on a test, or if I refer to something, I'm going to give you either SER or I'm going to tell you what each one means. Don't worry about that. If you know how to work the table, use the table, you'll be fine on any test question or anything that I ask. All right? It's not, don't stress about it. What I'm trying to point out to you here is that more than one codon can code for an amino acid. Right? Again, like for instance, you have leucine over here, LEUs, and I thought there was more than one, but I could be wrong. No, that's it. All right, but you can see where there's actually 6 codons that code for leucine. Now, you might be wondering what this stop is over here. Or what is the start? Well, like I said, DNA RNA is a language. It has an alphabet, U, C, A, G, if we're talking about RNA, U, C, G, and A, G. It has a vocabulary, words. That's what each of these things are. Each column is the list of words. And each of those words have meaning. What do they mean? They code for the different amino acids. Each word codes for an amino acid. There's only 20 amino acids, but the 64 words, or almost all of them, code for amino acid. Well, in our language, we have punctuation. How, when you're reading a book, how do you know that it's the start of a sentence? The letter's capitalized. How do you know it's the end of a sentence? There's a period or an exclamation mark. That's what these are. Whenever the ribosome reads the codon AUG, that tells it that it's going to start making the protein. It's the code that starts the protein, just like we capitalize the beginning of a sentence. Now, we can also stand for methanoalanine or something like that. I don't know, and I don't care. You don't need to know. All right. So we could see that start code in the sequence of codons in the amino acid, or in the sequence of RNA. So we might use the methanoalanine, or again whatever it is, in the amino acids, in the sequence of amino acids that we're making with the protein. But the first one will always be AUG. The first codon of the protein will always be AUG, because that tells the ribosome to start making a protein. Well, how do we know when we're at the end of a sentence when you're reading? There's a period. That's what these over here are. Notice that they don't code for anything except stop. So if one of those codons comes through the ribosome, it tells the ribosome, you're done making proteins. Don't make anything else. No more amino acids are going to be added to this protein. Stop. It's the period. It's the exclamation point. The ribosome is not going to be making any more of the protein. That's the stop code. And there are 3 stop codes. So you have 1 punctuation mark that says here's where you start a protein. You have 3 punctuation marks that say stop the protein. And everything in between, each codon codes for an amino acid, and it's brought in and put in sequence. So transfer RNA, tRNA, is what brings the amino acid to the ribosome based on the codon. So you've got this messenger RNA that's going through the ribosome, and the ribosome's reading codon by codon. It's reading the words, the codons. And transfer RNA is bringing in the correct amino acid to match that codon. Looks something like this. And I think this is a really good representation of kind of what it looks like, because the protein, the ribosome, is actually made of 2 subunits. And you can see there's 1 subunit here, 1 subunit there. You don't need to know that. I'm just saying this. I really like this diagram. All right, you have the different amino acids. You have 1 codon there, codon 3. So we break it up into 3. Break it up into 3. Here's this codon, which is I'm having trouble reading it. GUU, which you could go to the table. We could figure out what that was. I'm not going to do that. Well, actually, I am going to do that. Okay, we just go back. Here we go. GUU. So if we're looking at GUU right here, we're looking at G. So we're looking at down this one. GUU. Apparently that's valine. All right. So we know that this one right here is valine. We could figure out the other ones. I'm not going to do that. All right. And then you have a CUG. Okay, what's CUG? That one I am going to go back onto. And so you look at CUG. C is right here, and then U and G, so that's leucine. So we know that this right here is leucine. And what you're seeing is the transfer RNA. This is the transfer RNA, this little T-shaped thing, because that's actually apparently the shape of it. The transfer RNA right here, this, is bringing and holding the amino acid, reading this codon down here, and adding that amino acid to the growing chain of amino acids right there. The next one, which we know is leucine, is going to bring in, and it will pair up right here, and then this leucine will be added to this chain of amino acids. And this one right here, this next one, which you can see based on the codon, here's the codon that it pairs for, all right, is going to come in here, match up there, and this amino acid is going to be added to the chain. And then this one is going to be added to the chain. Proteins are just a string of amino acids determined by the sequence of bases in the DNA which form codons that the ribosome reads, and it just says, okay, this is the next amino acid we need. This is the next amino acid we need. And then you make this growing chain. This chain will continue to get longer and longer and longer as we keep adding amino acids to it to make the protein. So we have a start code, UAG. We have an end code, UGA, UAA, or UAG. Again, you do not need to memorize any of this. You will have access to a table on the tests. Just know how to use it. You got to know how to use it. That's all. All right. When we get to the end code, the stop code, the ribosome no longer adds any more amino acids. The protein sequence is done. It breaks off and goes and does its protein thing. Maybe it's modified in the endoplasmic reticulum, or maybe it's used by the cell for its own use. Here's a way to think about this. You go to a different country, let's say Italy, okay, or France, or Germany, someplace you don't speak the language. You have a friend with you who's you're staying with and is introducing you and showing you around, and you fall in love with someone from that country, but you don't speak the language, right? And you want to tell them you really care about them, that they're very important to you. So what do? You can't talk to them because they don't speak English and you don't speak their language. So what do? Well, you can tell them by writing a note. That is what transcription is. Transcription is changing it from DNA to RNA. When you write the note in English and you say, I really care about you, I want to see you again, or whatever you're going to write on your note to this person that you care about, all right? You're saying it in English. I care about you. I said it verbally. Then if I write it down, it's still in English, it's just in a different form. DNA to RNA is still in a nucleic acid. Just one is deoxyribose nucleic acid, the other one's ribonucleic acid. Still nucleic acids. It's the same language, it's just a different form. And that's what transcription is. If you think about when you transcribe something, you are taking what's being written and you are putting it into, you're being said and you're putting it into a written form. That's what transcription is. All right? So the messenger RNA takes that, your friend now takes that message that you've written down, your friend who knows where this person lives, will take that note to them. Again, in a written form, not in a spoken form. When they get it, they can't read it because they don't read English. The ribosome can't, there's no way of translating it. The ribosome is translating it, changing it from one language to another. The ribosome is going to read it and translate it into the new language. Non-English, Italian, French, German, whatever you need to have it translated into. That's translation. Translation's changing it from nucleic acids into amino acids. That's a whole other language. The ribosome is the translator. So you transcribe it, you change it from DNA to RNA, and then you take that RNA, that messenger RNA, to the ribosome, and the ribosome translates it into an amino acid, into a protein. It translates that sequence of bases into a sequence of amino acids. It's a different language. It's not, like it says, not in English. Now it's in amino acids, not nucleic acids. All right? That's how the cell produces proteins. You read the DNA, base pair by base pair, unzip it, just copy it over to messenger RNA. Instead of thymine, we use uracil. We take that messenger RNA, it can leave the nucleus, go out to the ribosome, and translate it into amino acids, into a sequence of amino acids, which becomes a protein. So let's take a look at what we know. All right. Watson and Crick knew it had to be able to store information, copy itself, make mistakes, and be able to be read. We've talked about how it stores information. Well, let's come back to that. We've talked about how it replicates itself. Unzip itself, complementary strands, A's with T's, G's with C's. All right. We've talked about how it can make mistakes. Sometimes when it copies itself or it corrects itself, it doesn't do it correctly. It forces something that is not designed to fit there to fit. How do we read it? We read it in the sequence of base pairs. That sequence of A's, T's, G's, and C's becomes the sequence of A's, U's, G's, and C's in RNA. And the RNA goes out to the ribosome where it's translated into amino acids. That is how the whole sequence of DNA works. The whole molecule works. Again, everybody, like when we started the unit, you were like, "Oh, this is it makes you, and it makes..." No, it doesn't. All it does is code for proteins. Those proteins, those enzymes, all the other proteins determine who you are, what you are. The sequence is different in a cockroach. The DNA is different in a cockroach than it is in you. Therefore, it's going to make different proteins. Those proteins will interact differently, and they're going to make it in a cockroach, whereas you're a human being, I hope. If you have any questions, please make sure you ask in class or an Edpuzzle. I hope you took good notes. Good luck.